Hi, so far we've been talking about the effects of neurotransmitters in general, but now we're going to start talking about specific neurotransmitters and their properties because different neurotransmitters do different things. So let's go ahead and dive in. All right, so one of the most fundamental differences is that some neurotransmitters are what are called small molecule neurotransmitters, and others are called peptide neurotransmitters or neuropeptides, and they're different in terms of their chemical structure, how and where they're synthesized, how they're processed, and how they're released. And we talked a little bit about their release when we talked about the different types of vesicles, so we'll be revisiting that again. We're mainly going to focus on the small molecule and the peptide neurotransmitters, but I'm also going to talk briefly about the retrograde neurotransmitters, which is something that you'll want to know about if you continue in neuroscience. So, the main types of neurotransmitters are small molecule neurotransmitters. These handle basic moment-to-moment communication. They are the workhorse neurotransmitters in the brain. They're used all the time and they're used for basic communication between neurons. A subtype of these are the biogenic amines. These handle neuronal function. They modulate neuronal function and they indicate kind of overall brain states. In contrast, the neuropeptides, well, they also modulate neuronal function, but they handle each one handles specific tasks. So there are a great many different neuropeptides used in the brain, and they each have a specific job. Retrograde neurotransmitters are interesting because they signal backwards from the postsynaptic to the presynaptic cell. So that's backwards relative to what we've been talking about at chemical synapses. I'll talk a little bit more about that. So the small molecule neurotransmitters are called that because they are small. They are roughly the size of an amino acid. And because they are small, they can be synthesized on site by enzymes in the axon terminal, the presynaptic terminal. And they are transported into vesicles. So by vesicular transporters that are also in the presynaptic terminal. These vesicles are made by the vesicle recycling process that we described earlier. And the vesicles that have these are these small clear core vesicles, and they can be released with a single action potential because they are part of the um, pool that is ready to be released, the readily releasable pool. And these three in particular, acetylcholine, glutamate, and GABA, are ones seen over and over and over again when you talk about brain function. And we'll be talking about those further in further videos. The biogenic amines are a special subset of small molecule neurotransmitters. They are also small, roughly the size of an amino acid. And you can see their chemical structures here of the main types. They, similar to other small molecules, they are synthesized by enzymes in the presynaptic terminal and transported into vesicles by vesicular transporters that are in the presynaptic terminal. However, these are dense core vesicles. They form small, dense core vesicles. So they're going to be further away from the membrane and thus require more calcium and thus sustained firing before being released. In contrast are our neuropeptides or our peptide neurotransmitters. And as I said, there are like hundreds of these. These are four interesting ones. Why are they called that? Neuropeptides, because they are peptides. So unlike the ones we've seen previously that are about the size of a single amino acid, and glutamate just straight up is the amino acid. These are chains of several amino acids put together. And they all have different jobs. So like beta endorphin is used for alleviating pain. It's an endorphin, like an opioid. Substance P, on the other hand, is used to signal that there is pain. Orexin is used in the kind of awakeness system, and GLP-1, I included that because that's what these new injectable weight loss drugs like Ozempic mimic. So it has to do with regulating appetite. So this is just like a flavor of some of the neuropeptides that are out there. Now, these, as you can see, are larger in size And because they are peptides, they are synthesized just like proteins. So everything you know about protein synthesis is true of making these neuropeptides. And so thus they are made by ribosomes that are in the cell body. And where they're packaged into vesicles is also in the cell body. So they're packaged into vesicles by the Golgi apparatus. And those vesicles are then transported down the axon to the axon terminal, um, just like we talked about earlier about axonal transport. These are going to be large, dense core vesicles. And again, they're going to be further away from the membrane and require more calcium and thus sustained firing for release. Final category is our retrograde transmitters. These signal backwards from the postsynaptic to the presynaptic cell. They are made postsynaptically then, and they spread through the cell membrane. They diffuse through the cell membrane. They tend to be lipid soluble into neighboring neurons. So one example here are endocannabinoids. If that sounds familiar, it's because they're in cannabis and marijuana. And these are endogenous molecules that target cannabinoid receptors, which is the same kind of receptors as the psychoactive substances in cannabis. As you'd expect from observing people who are high on weed, endocannabinoid signaling is important for perception, motor control, memory, and appetite. All right, that's all, and I hope that was helpful.